So in this video, we're going to be talking about the membrane potential, and action potential, and how this triggers a signal sent down the neuron, and what ultimately triggers these responses. So first, a few terms. The membrane potential is the difference in electrical charge between the inside and the outside of the membrane. Membranes will have a slightly positive charge on their outside and slightly negative charge on their inside. This difference in membrane potential ultimately is what causes the membrane potential, or what defines the membrane potential. An excitable membrane is a membrane that can change its membrane potential when stimulated. There's two types of cells in your bodies that have excitable membranes. One are neurons, which was the focus of this discussion, and the other is muscle cells. An action potential is then the brief change in the resting membrane potential. All right, so to understand membrane potential, we have to understand a few things related to what determines the flow of energy and the potential energy. So the flow of ions in the body is known as the current. Current is equal to voltage over resistance. So voltage is a measure of potential energy and is the difference in positive and negative charges. So the membrane potential represents a voltage because it is the difference in membrane charge. Resistance, in the case of cells, is what is provided by the cell membrane. It's essentially the tendency of a substance to hinder the flow of electrons. So the membrane has a tendency to resist the flow of things across the membrane. Now, what leads this to having a membrane potential, it has to do with the solutes, specifically the anions and cations that are dissolved in cell. So in the extracellular fluid, we have a slightly higher concentration of sodium. Sodium has a positive charge, and this leads to a partial or a more positive charge on the outside of the membrane. Potassium is higher inside the cell, but it's overall going to be negative because anions are going to be higher inside the cell compared to outside the cell. So this overall gives a negative charge or a net negative charge on the outside. So the rest are on the inside of the membrane. So the resting membrane potential, that is the steady state potential, is going to be around negative 70 millivolts for neurons. In order to maintain this, we have to have a flow of sodium and potassium across the ions. So our sodium ions are higher on the outside of the cell, and our potassium ions are higher inside the cell. What maintains this is what is known as a sodium-potassium ATPase pump. So the ATPase pump is going to utilize ATP. When it utilizes the ATP, it's going to hydrolyze it into ADP, and this in turn will release an inorganic phosphate. Notice that the sodium the channel is going to be open to sodium binding on the inside of the membrane. It binds, then you have ATP binding in, going through hydrolysis. This changes the conformation of the protein so that the channel now opens to the extracellular space, and it releases the sodium ions. Now you have a phosphate bound to the sodium potassium ATPase. Potassium binds to it. When the inorganic phosphate is released, then you have a change in conformation, and it opens up to the inside and releases the potassium ions. And so this helps maintain that high concentration of sodium outside and high concentration of potassium inside. In addition to the sodium potassium ATPase pump, there are going to be ion channels that are going to uh, control whether things are going through the membrane. The ion channels tend to be specific to um, the ion that it's letting through. So this can be due to the size, the charge of the particle. And then what else determines the whether something will move through the ion channel is its state, whether it's open or closed. Overall, when the channel is open, ions passing through will follow a chemical. That means that it's going from high concentration to low concentration. And an electrical, that means it's going from positive to negative charge. So positive tends to be attracted by a negative charge. There are different types of ion channels. The ones that we are going to focus on are going to be leakage, ligand gated, mechanical, and voltage. So first, leakage channels are ion channels that remain open, or they may open randomly. They're not gated. These are going to just help maintain membrane potential. Ions move down their concentration gradient when these leakage channels are open, and we see this type of channel being significant for potassium, sodium, and chloride channels. The next type are the ligand gated, also known as chemically gated ion channels or ionotropic receptors. These open when a chemical that is specific to the channel binds to it. So you might have a neurotransmitter like acetylcholine bind to it. When acetylcholine binds, it opens up the channel. So no binding, the channel is closed. When the channel binds, or when the neurotransmitter binds, channel opens, and ions that are the right charge and small enough to go through the ion channel go through down their concentration gradients. Mechanically gated ion channels are similar to ligand gated channels, except they're going to be sensitive to some sort of physical deformation. So it could be pressure or some other mechanical force that ultimately leads to the channel being open. So touch, pressure, receptors, these fall into these categories and are what's going to trigger the opening of these channels. And then finally, we have voltage gated ion channels. These are going to open and close due to a change in the membrane potential. So negative 70 millivolts, you can see that this ion channel is closed. It has this blocking the exchange of things going through it. Change the voltage to negative 50 millivolts, and this opens up, and now the ions can move down their concentration gradient. We're going to look at the membrane potential of a neuron as it gets stimulated and has a change in the action potential. So we start off with our resting membrane potential of negative 70 millivolts. At some point, a stimulus, such as a chemically gated or mechanically gated ion channel, triggers a change in membrane potential. So this could open up and allow sodium, which is higher outside of the cell, to flow into the cell, making the cell membrane less negative and more positive. Eventually, it meets a threshold of about negative 55 millivolts. That voltage threshold triggers the opening of voltage gated sodium channels. So now sodium continues to flow into the cell, making it less negative and more positive because you have positive charge moving inward. This change in membrane potential is known as membrane depolarization because you're depolarizing the difference between them. Ultimately, you overshoot zero and go to about positive 30 millivolts. This is when voltage-gated sodium channels will close, so sodium can no longer flow into the cell, but voltage-gated potassium channels open, meaning potassium can flow out of the cell. So now you have a positive charge going out of the cell, making the inside of the membrane more negative, leading to membrane polarization as the potassium flows through the voltage-gated ion channels. This is going to overshoot our negative 70 millivolts, and so you have what is known as hyperpolarization. This happens because some of the potassium channels remain open while the sodium channels are reset, and then eventually this is going to equalize and you'll get back to your normal resting potential. All right, one thing to make a note of is our sodium or voltage-gated sodium channels have three different conformation states. It can have a closed state, an open state, and an inactivated state. So the resting membrane potential has it in the closed state. So you can see closed has no passageway through it. Open, you have the passageway. Inactivated, you have the passageway, but the passageway is blocked. So starts off with a closed conformation. The closed conformation remains until it reaches the trigger where it opens. When it opens, it is going to allow things to go through until it reaches about positive 30 millivolts, at which point it's going to become inactivated. Inactivated is going to remain that way initially, but it'll resume its closed potential as the membrane equalizes. What's important about this is that you have potassium channels closed in this region while the potassium channels are open in this region, meaning that regardless of the strength, when a patch of membrane is generating an action potential and voltage gated channels are open, the neuron cannot generate another action potential. However, when most sodium channels have returned to the resting state, but some potassium channels are open and repolarization is occurring, the axon's threshold for AP generation is elevated, leading to a strong signal that can be overcome. So these are going to be the absolute and refractory or relative refractory periods. Absolute, when you absolutely cannot initiate another action potential. Relative, when it's going to take more of a stimulus to cause an action potential than you would normally get. All right, so let's look at what's going on at the axon terminal and the synaptic gap as the membrane potential moves down. So the axon of the presynaptic neuron has voltage-gated calcium channels. It also contains synaptic vesicles with neurotransmitters, the synapse, and then you have your postsynaptic neuron. This postsynaptic neuron has ligand-gated sodium channels that can let things go through. So as the membrane potential goes down, the voltage-gated, so you have membrane potential going down the axon terminal. As the action potential reaches the voltage-gated calcium channels, they are going to open and calcium is going to come into the cell. This is going to trigger synaptic vesicle fusing with the membrane, causing the neurotransmitters to diffuse across the synaptic space, interacting with the ligand-gated sodium channels, which then open and allow sodium to enter the dendrite of the postsynaptic neuron. This ultimately is what is going to trigger the movement of the action potential along one synapse to another. All right, that does it for the introductory stuff on the neurons and nervous system. We're going to start looking at the central nervous system next. I'll see you in the next video.